Mendel's results didn't actually receive much notice during his lifetime. It wasn't until 1900, about 16 years after his death, that three different investigators independently rediscovered his pioneering paper. They came across it while searching the literature in preparation for publishing their own findings as information that they needed to put in the background section of their papers. It was after this point of rediscovery of Mendel's ideas that many investigators set out to test them. However, scientists attempting to confirm Mendel's theory often had trouble obtaining the simple ratios that he had reported. That's because he had separated out these 7 traits and known that they were absolutely true breeding and they were dominant and recessive. Black and white, so to speak. Most traits aren't really that way. A number of assumptions were built into Mendel's model that are oversimplifications of how heredity works. But it's a great model to begin from. These assumptions include that each trait is specified by a single gene, and we know that's not really the case. Each gene has only 2 alleles, and we also know that there are many different varieties of, let's say, for example, hair color, and that there's a clear dominant-recessive relationship between the alleles. And as you'll see, that's not always the case. So most genes don't actually meet these criteria, and so we need to look a little bit beyond Mendel's ideas to understand some more concepts of heredity. Let's look, for example, at polygenic inheritance. Polygenic inheritance occurs when multiple genes are involved in controlling a phenotype or a trait. You can often tell that something's polygenic because it occurs in a spectrum, for example, skin color or height. There isn't just tall or short. There's all kinds of heights and all kinds of skin colors. So the traits show continuous variation that you can see down in this histogram below about height. Pleiotropy is another idea. It refers to an allele that has more than one effect on the phenotype. Pleiotropic effects are difficult to predict because a gene that affects one trait often performs other unknown functions. A great example of pleiotropy is in cystic fibrosis or sickle cell anemia. There are multiple symptoms, for example, in sickle cell anemia that can be tracked back to one defective allele, and that causes the blood cell to sickle. And then those sickled blood cells stick into the vessels and clog them and cause many, many effects that we see in the sickle cell anemia disease. So pleiotropy is when genes have more than one effect on the phenotype. And polygenic inheritance occurs when multiple genes are controlling one trait. Now let's consider multiple alleles. There's often more than 2 alleles for a gene in a population. The ABO blood typing in humans is a great example. There are 3 alleles. There's the A allele, the B allele, and the O allele. Each individual, though, can only have 2 of those alleles. So you might be type AB, or you might be type AA, or BB, or AO, or so on and so forth. The number of alleles possible for any gene is constrained, but usually more than 2 alleles exist for any gene in an outbreeding population. Another concept is incomplete dominance. Incomplete dominance is where the heterozygote is an intermediate of a phenotype that's in between the 2 ends. For example, here, when we look in snapdragons, we have red snapdragons, white snapdragons, and we get pink by crossing red and white. So the heterozygote is actually an intermediate of both homozygotes. And then there's codominance, in which the heterozygote shows an aspect of both phenotypes from both homozygotes. For example, type AB blood. Both A and B are dominant alleles to O, which is recessive. Let's look a little closer at this ABO blood grouping. It's a neat system and it demonstrates both multiple alleles as well as codominance. As we've already discussed, there's 3 alleles for the gene. We have IA, IB, and then a lowercase I, which would be the O genotype. Codominance is where IA and IB are both dominant. That is, the blood cells express the antigen A as well as the antigen B. It doesn't blend out into something in between A and B. Both phenotypes are expressed. In this table below, you can see this more clearly. Blood type A has a cell label that's called galactosamine, and blood type B has galactose on the cell surface as a marker. However, type A and B has both galactose and galactosamine, which is a whole novel phenotype because it's expressing both genotypes. And then II, or blood type O, has no cell labels on it at all. The environment also has a big influence on phenotype. The coat color, for example, in Himalayan rabbits or Siamese cats is influenced by the environment. I don't know if anyone has a Siamese cat, but you'll notice if they're more of an outdoor cat, they're a darker color all the way over. And if you have an indoor Siamese cat, then they're much lighter in color. The allele that's responsible for coat color in these animals produces an enzyme that allows pigment production only at temperatures below 30 degrees C. So the more central regions of the cat are always above 30 degrees C, and so they don't produce any pigment at all, so they're white in color. However, the tips of the ears and the tail and the legs get a little bit more exposure to temperature in the face, and so they will exhibit the pigmentation. So in these cats that spend more time outdoors, you'll see darker pigmentation and larger areas of pigmentation. So this is a clear case where the environment influences the expression of a gene. One of the assumptions in Mendel's model is that products of genes do not interact. But as it turns out, the products of many genes do interact. For example, the production of pigment may involve a multi-step pathway, as you can see here. So epistasis is a situation where the behavior of gene products can change the ratio expected by independent assortment, even if the genes are on different chromosomes that exhibit independent assortment. In this case, we're looking at corn. And a researcher named R.A. Emerson crossed two white varieties of corn and found that the entire F1 generation was purple. And that in the F2, we didn't have the normal 9:3 to 3:1 ratios we might expect. But as it turns out, the production of this purple pigment is a two-step process involving two enzymes, enzyme A and enzyme B. In order to actually get the production of purple pigment, a plant must possess at least one functional copy of each enzyme's gene. So that would be the capital version, the dominant version. If it lacks either one of these in the dominant form, then there'll simply be no color produced. So we have to have a capital A at least and a capital B at least in order to get the purple color. If we only have the recessive or the mutant form, then enzyme A doesn't work to help produce purple, and nor does enzyme B. So we can perform this cross in just the same way. We're crossing the white plant and another white plant. Both have one functional enzyme, but not the other, so they cannot produce the purple pigment. However, when we cross these two plants, we find that they're heterozygous for both enzymes and produce purple pigment. When we self the F1 generation, we'll see that we get the same genotype ratios as we expect. 16 different genotypes. However, we don't see the expected ratios that Mendel noted. We get 9 purple ones and 7 white ones. The 9 purple ones all have at least one big A and one big B in order to produce the purple color. All the others that are white lack either a big A or a big B or both, and thus they don't produce any purple pigment. So in short, epistasis is one gene affecting the expression of another gene. In Labrador retrievers, we also see epistasis. Consider these two alleles. B is the color allele. If we have big B, we have a black lab. If we have big B, we have a brown lab. However, there's an additional gene in the creation of pigment. We'll just call it the color gene. If we have a big C, then we're allowed color. However, if we have only a little c, we get no color. You have to have at least one big C in order to get color. So let's consider labs that are heterozygous in both traits. We have big B times big C crossed with another brown lab. that also has color permissions. When we look at the genes that could be produced by, when we look at the gametes that can be produced by these, we can get a big B, big C. We can get a big B, little c. We can get a little b, a big C, and a little b, little c. We'll draw ourselves a nice Punnett square. And put the same gametes along the other side, because after all, both parents will produce the same gametes. When we fill out the Punnett square, we'll see the same genotypes that we would expect in a Mendelian cross. Oh, brown was the recessive trait. Let's correct that. So little b equals brown. So now let's analyze our results. Remember that in order to have color, we have to have at least one big C. Here we have a black lab and it's allowed color. So there's one black lab, a black lab and it's allowed color. Heterozygous black with color, heterozygous black with color, black with color, no color, black with color, black with no color, black with color, black with color, brown with color, brown with color, black with color, black with no color, brown with color, and brown with no color. So in this case, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 black labs. We have 3 brown labs and 4 with no colors, so they would be yellow labs. So a 9:4:3 ratio. Again, the whole point here is that one allele is masking the effect of another. If we don't have permission for color, as in all little c's, then no color can be produced. If we do have permission for color, having one big C, then we'll see that black and brown show through. So that brings us to the end of our chapter on inheritance. Let's take a quick card quiz. When 2 alternate forms of an allele contribute equally to a phenotype, it's called a trait affected by multiple genes is when one gene masks the effect of another gene, the ratio is modified. This phenomenon is in Labrador retrievers, gene E codes for the presence of pigment and little e for no pigment. Gene B codes for the amount of pigment deposited. B for black, little b for brown. Which individual below would have a yellow coat? In snapdragon flowers, red is dominant and white is recessive. What color would a snapdragon with the genotype big T, little t have if flower color was incompletely dominant? Here are your answers.